This is the main lecture of the series of videos on fine mapping. So if you have chosen to skip the last two lectures, that's okay. They're optional and they're not required to understand this video. So without having met my audience, I assume that there are a variety of level of fluency in fine mapping among you, and you may have very different goals coming into this class. Some of you may just want to know what fine mapping is and see if it can benefit your research. Some of you might be preparing to run fine mapping on your dataset, or maybe you need to interpret fine mapping results and make decisions about follow-up studies. Or you may need to debug weird results or go into fine mapping method development like me. For all of you, I'm hoping this content of the video will be helpful. This lecture is split into two parts. I will start by quickly reviewing the goal of fine mapping and show you a few fine mapping results in real data. And then I'll talk about many different factors that influence fine mapping resolution and efficacy. And we should be aware of these factors when applying methods and reading results. So those two bullet points are going to be in the first video. And then the second part, I will go through current methods, give you an overview of the methods and how they work. And then I'll talk briefly about multi-cohort fine mapping, its motivation, and some of the standard approaches. So first, let's review the goal of fine mapping. In the first video, I talked about what is fine mapping, and I contrasted it with GWAS. They have very different goals, but they're related. So a GWAS scan it's scanning the genome for any sign of association. And the goal is discovery, but not causal inference. Association is not necessarily causation. And if you zoom in on one locus, you can see that there are many, many SNPs that are above the genome-wide significance threshold right here, purely because they are in LD with the lead SNP, not because any of them are necessarily causal. So suppose we have two causal SNPs in the region. This is unobserved ground truth. You don't know this. And the GWAS result for this region that you can see will contain many significant SNPs. But most of these are only significant, like I was saying, because they have non-trivial LD with either or both of the causal SNPs. And fine mapping, its aim is to recover this ground truth and identify likely causal variants. So how does fine mapping do this? How does fine mapping quantify causality? It's through two concepts. We will focus on Bayesian fine mapping methods in this lecture because they are more commonly applied. So fine mapping outputs two quantities. One is called posterior inclusion probability, and it is the probability that a SNP is causal given the observed data. Every SNP will be assigned a PIP in the typical fine mapping method. So in this plot, you can see that PIP is plotted on the y-axis. And there are some high PIP SNPs, there are some low PIP SNPs. And the second output for a typical Bayesian fine mapping method is credible sets. And these capture the uncertainty of the identity of the causal SNP due to LD. For example, in this 5 SNP set, the algorithm is basically telling you that it knows that there is a good chance a causal SNP is in there, but it is uncertain which one of the 5 it is. Now, let's take a look at a few genomic regions and the fine mapping results generated on those regions. So these fine mapping results are based on the paper by PGC in 2022, where they GWASed and fine mapped the phenotype schizophrenia. Let's first look at a simpler region on chromosome 7, harboring the gene GRM3, the glutamate metabotropic receptor 3. This is a key regulator in glutamatergic neurotransmission in the brain. So the GWAS signal here is fairly strong. There are about 2 dozen SNPs passing the genome-wide significance threshold, but we would really like to narrow down the candidate causal SNPs among the 2 dozen. And this is where phi mapping comes in. So after applying phi mapping in this region, I have this PIP plot for this region. 2 SNPs stand out. Their PIPs are around 0.5, and they are colored purple, the same color indicating that they are in the same credible set, which usually means that they are highly correlated and the algorithm can't really tell which one is the true causal one, but it knows one of them is. There is another credible set in this region. It's colored yellow. That's indicating that this region may have more than one independent causal signals. But the algorithm is really having a lot of trouble with this yellow credible set here. You can see the credible set size is quite large, and none of the SNPs in there have gotten very impressive PIPs. The highest is about 0.2. So perhaps that yellow signal is not going to get teased out now, but in the future, maybe if we increase the sample size for this phenotype, we have a better chance of nailing down exactly which one of these SNPs is the causal one in the yellow credible set. Now let's look at another example. So this one is a little bit more complicated than the previous one. Right off the bat, you know that there's probably more than one causal signal in this region because there are 2 peaks. And applying phi mapping, we actually see 3 credible sets. So 3 different colors in this plot, if you can see it. What's interesting to note is that this highest PIP variant, this RS131 variant, is actually this variant C down here. And it's not one of the highest significant variants in this region. And the reason that this SNP got a high PIP is not because that is somehow more causal than the rest of them. And the reason is mostly because the SNP has less LD friends. Therefore, there's less competition for PIPs. And SNP A and SNP B here have more LD friends, so they have to share their PIPs, and it's harder for the algorithm to pinpoint which one among all of these highly correlated variants are the causal ones. So one last example, and this is a complicated one. You can see that many variants here have correlation almost equal to 1. Red means the LD is close to 1. And this is going to be hard for phi mapping algorithms. And not surprisingly, after we apply phi mapping, this credible set is very large, and none of the SNPs have PIP even passing 0.1. The biggest one is 0.05. So now you have an intuitive sense of what kind of region is easier to phi map and which ones are harder to phi map. Let's learn more comprehensively what factors can influence phi mapping efficacy. Efficacy, I'm referring to accuracy. Maybe I should have just said accuracy. That is whether or not phi mapping can actually include the causal variant in its credible set. And also phi mapping resolution, which refer to the size of the credible set. Let's first look at LD and sample size. In this very useful paper that I recommend all of you to read, is Shade et al., 2018, Nature Review Genetics. He did a simulation. So he simulated a region with 5 SNPs in it, and only one is causal. And in his simulations, he set different levels of correlation that's indicated by this row here. And he also added the sample size factor. So he simulated different levels of sample sizes ranging from 500 individuals to 20,000 individuals. Then he ran phi mapping in all of these simulations. And then he looked at the PIP of the causal SNP. Because he did the simulation, he knew which exact SNP is the causal SNP, so he can look at that. And he noticed that as the correlation among the SNPs in the region increase, the causal SNP's PIP is going down. And the algorithm become less and less certain about the causality of the SNPs. But then you notice that this can be compensated by higher sample size. So as you go up in sample size, the PIP drop gets slower and it's more delayed. For this 20,000 sample size, it's not until the LD goes above 0.99, 98, the causal SNPs PIP start to drop from 1. And the message is this: more sample size and less LD means easier fine mapping. So here are some prominent fine mapping applications in different data sets, just to give you a sense of what kind of sample size is out there for fine mapping applications. And usually in biobanks and consortia, 100,000 individuals is a good sample size. Bigger data sets like FinGen and the Million Veteran Program has more than 500,000 individuals, and it's still growing. For some cohorts, especially Finland, where the LD in that population is generally higher due to population bottleneck, a larger sample size is actually needed in order to combat the high LD. And aside from sample size and LD, we notice here that SNP density is a factor that can influence fine mapping resolution. And when there's more SNPs in the region, so now not only 5 SNPs here, there's 10 SNPs and 20 SNPs and 40 SNPs. So when there are more SNPs in the region, the influence of LD on fine mapping resolution gets amplified. And if all other factors are equal, a region with less SNPs should be easier to fine map than the region with more SNPs. There are some other important factors that fine mapping is sensitive to. And first and foremost, missing causal variants. If the causal variants are QC'd out for some reason, or it's just not genotyped or imputed, then all you left are its LD friends who are forced to try and explain the signal. In that case, you will get false positives. And the second is effect size of the causal SNP. The higher it is, typically the easier it is to identify it as causal. The third one is the number of causal SNPs. As the number of independent signals increase in a region, it becomes harder and harder for the algorithm to tease them apart. All right, so that's all for the factors influencing fine mapping. In the next part of the lecture, I will start with an overview of fine mapping methods.